Hi everyone, this is me, obviously your biology professor, Kelly Roberts. So we're going to do something a little bit different for our review this time, since in the interest of time, once again, we won't be able to have a face-to-face review, and in addition that this is really heavy material, so there is a lot of really meaty stuff in this exam. In fact, this is probably going to be one of your most difficult, actually it's going to be your most difficult exam here in microbiology. So to try to give you all a bit of a more guided study discussion, I'm going to point out some things that you really need to make sure you know. Notice how I am stressing that. I'm actually making this recording at 1:21 a.m. That's how frickin' important this is, that you know this information. I don't really even have like anything like cool to show you. I guess I'll share my desktop with you so that we can look at something other than a white page. Okay, so what I am sharing with you here is a document that you have on Blackboard. It's in the exam review file. If you're going over this and the PowerPoint, you're in a really good position. And if you're going over those flipped activities that we've done, you're in a really good position. But I'm just going to give you this added information to sort of help you understand where I want you to focus your studying on for this next exam. All right, so things that I want you to make sure that you really understand is the difference between an anabolic reaction and a catabolic reaction. An anabolic reaction is going to allow for things to be built up, and a catabolic reaction is going to allow for things to be broken down. So metabolism is the study, and that's actually right here at number 6. Metabolism is the study of all of those catabolic and anabolic reactions. So make sure you understand anabolic, building things up, energy goes into that process. Catabolic, breaking things apart, energy goes out. Make sure that you understand who the final electron acceptor is in both aerobic respiration and anaerobic respiration. In aerobic respiration, that final electron acceptor is oxygen, and in anaerobic respiration, that final electron acceptor is just an oxygen-containing compound. And I'm trying to find one of those questions on there. I feel like I have one, and maybe I don't. But even if I don't, you need to know that. So nitrate, sulfate, carbonate, those are all great examples of that. Also make sure that you understand the parts of enzymatic activity. So know that an enzyme has, a whole enzyme has its protein portion and its non-protein portion, and also make sure that you understand that an enzyme works on this entity, whether it breaks it down or builds it up, and that entity is called a substrate. So that's that molecule or something that it is acting upon. I also want you all to make sure that you understand the difference between competitive inhibition and non-competitive inhibition. In class, we looked at one of the videos that showed how sulfonamide can actually mimic the shape of a substrate that's made by a bacteria, and what was that as an example of, competitive or non-competitive inhibition. In non-competitive inhibition, remember that the molecule will bind to an allosteric site, so some area outside of the active site, and the active site is where the substrate will bind in non-competitive inhibition, but it still can changes the shape of the active site, so the substrate that's normally supposed to be worked on doesn't get worked on, so make sure you understand that. Also make sure you understand what goes in and out of each of those 3 steps of cellular respiration, both aerobic and anaerobic cellular respiration. So if we start, what goes into glycolysis is glucose. What comes out of it is pyruvate. You also have NADH that's made that comes out of it, and you have ATP that comes out of it. So understand what that's a product of. In the Krebs cycle, we have that pyruvate that gets groomed, so it becomes coenzyme A through that whole grooming process, losing CO2, and then also generating an NADH. Remember, all those NADHs go to the electron transport chain, and they what goes into the Krebs cycle is that groom, so now that it's been broken down, that pyruvate is no longer pyruvate, it's coenzyme A. It goes into the Krebs cycle. That happens twice. And what comes out of the Krebs cycle is carbon dioxide, you get ATP, you get FADH, too, and you get NADH. So make sure you know all of that. Also, make sure that you know how fermentation works. Specifically, make sure that you know that the final electron acceptor in fermentation is an organic molecule. Also understand where the process of glycolysis, the Krebs cycle, and the electron transport chain takes place in prokaryotic cells, and that's the Krebs cycle takes place in the cytoplasm, glycolysis takes place in the cytoplasm, and then along the cytoplasmic membrane is where we have most of the ATP made, because that's where the electron transport chain happens. Understand redox reactions, that reduction is gaining electrons and oxidation is losing electrons. So make sure you understand that. Make sure you understand those questions that come off of the worksheets that we've done, those flipped activities. There's 3 worksheets that we have for chapters 5, 6, and 7. Make sure you go over all of those. There are some of those questions that are kind of tricky, like why does FADH2 yield less ATP than NADH2? And that's because the FADH2 electrons enter that ETC chain at a lower energy level. So make sure you understand that. Also understand those 2 different classes of fermentation we had. We had lactic acid fermentation and alcohol fermentation, and understand in that lactic acid fermentation that that's what you got as a byproduct, lactic acid, and then what you get as a byproduct of alcohol fermentation is ethanol alcohol. Also know how many NADHs and FADH2s you're going to have leave out of the Krebs cycle. So make sure you know that information. Make sure you understand that we can call a bacterial chromosome. The bacterial chromosome can be both circular, that we call a plasmid, and it can also be found in that nucleoid region, so make sure you know that. Make sure that you understand that AUG is always the start codon. Understand how the semi-conservative DNA replication means that we have one old strand and one new strand. So the daughter strands are going to consist of the original strand and then one new strand on there. Also understand this idea of silent mutation. So if we don't change if the amino acid sequence is changed, that's fine. But if both the old amino acid sequence like AAA and then the new amino acid sequence like AAG, if they both code for the same amino acid, let's just say nucleotide sequence, not amino acid sequence, but if the nucleotide sequence is slightly altered, but we still get the same amino acid, then it's considered a silent mutation. So make sure you understand that. Make sure you understand horizontal gene transfer as opposed to vertical gene transfer. In horizontal gene transfer, this is where we're going to have bacteria in the same generation are going to be changed in horizontal gene transfer. In vertical gene transfer, that's just an example of binary fission. We have 3 different types of horizontal gene transfer. Transduction, which involved a virus known as a phage. We had transformation, where we had competent cells that could take up DNA from the external environment. That was Frederick Griffin's experiments. And then we also had conjugation, which was what we call bacterial sex, where it happened through a sex pilus, and one cell gave genetic information to a donor cell, gave genetic information to a recipient cell. So make sure you understand those horizontal gene transfers. Make sure you understand that a Lac Operon is considered an inducible operon, and know all the parts that go with either an inducible or repressible operon. So you need your operator, you need your structural enzymes, and you have repressors. Like, those are all things that are part of this operon system. Understand that if you just transfer a random piece of DNA by a phage or a virus, that we just call that kind of generalized transduction. Make sure you understand what happens in transcription. So in transcription, we are going to make sure that we get the correct portion of the DNA, because the DNA is used as a template for copying. Make sure we get the correct portion of the DNA, and we have and to make our messenger RNA. So the DNA, remember, is a crime boss, and it's sending the message to make the messenger RNA in transcription. So make sure you understand that. Make sure that you understand that the ribosomes, the DNA polymerase, the RNA polymerase, they're always going to move along the strand in a 5' to 3' direction. Understand that repressible operons are always turned on unless they are turned off, like something happens to turn them off, and inducible operons are always turned off unless something turns them on. So make sure you understand that process. Also make sure that you understand how to code from DNA to messenger RNA to amino acid sequence, and also make sure you can go backwards from that, so that if I gave you the amino acids and their corresponding codon for the messenger RNA strand, that you could figure out what was on the DNA strand. So since we know there's no uracil in DNA, then anything that's got a U in it, we automatically mix that off. So make sure you can go forward and you can go backwards. So I'm going to go ahead and I'm going to stop sharing. Let's see if I can find it. All right, so we have kind of a, it's not really going to work. So you have this question here at 37. If you can go forward, then you can definitely go backwards here. And what I would challenge you all to do is that I would challenge you to go ahead and figure out what this protein is, and then cover that answer up, and then go back and see if you can make the anticodon. And then go back and see if you can make the messenger RNA. So for example, I'm just going to do a very short one here that I'm going to type at the bottom, that if I give you the amino acid sequence of serine and alanine, and let's do one more, glycine. If I gave you that amino acid sequence and asked you to make the corresponding DNA sequence, it just kind of popped up on the other side, so I'm just going to shorten this a bit. Slate. Okay, so if I give you a corresponding amino acid sequence, and I would have to give you a chart to show, you know, what nucleotides go with those things. So I'm just going to tell you now that for serine, what that amino acid sequence is, let me find it first. It can be UCU or UCG. And then for alanine, it is GCA. And then for our third one here, which we have as lysine, lysine is AAG. So now I technically have, I'm just going to delete some more space here. So I technically have my messenger RNA strand here. So this right here is my messenger RNA strand. It's highlighted. Now from there, I want to make my DNA strand. So U's partner is going to be A on that. So U to A. And then G's partner is still going to be C. U's partner is still going to be A. And then U's partner again will be A. Well, actually, I don't have to do that one because that was just a copy. G's partner is C. When I say it was a copy, like these two both are for serine. And so I have to go to this next one now. So G's partner is C. C's partner is G. And then instead of putting U's here, because we're making DNA, then we have T, 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 and C. So if I had this as a multiple choice question, I would do it this way, and I would also do it where instead of using UGU, because remember that was one of the other options for serine, that I would just use UCG. So I'm not going to make the whole thing over again because just the first set of letters is going to change. Then I'd have A, C, A, G, C, and then the rest of this would all stay the same. So that would all stay the same. All right, so really what I wanted you to get from that, and if that didn't make any sense, please stop this video and rewind it to hear it again. But I want to make sure that you can go from DNA to messenger RNA to protein, which is at question number 37, and I also want to make sure that you can go backwards from protein or amino acid sequence to messenger RNA to DNA. So that's what that whole question was about. All righty, let's see what else we have here. Make sure you understand that conjugation is not reproduction nor is it sex, because we don't get any offspring as a result from it. We just get a genetically changed bacteria as a result. Make sure that you understand those different, we had a question on this on your Chapter 6 worksheets where we talked about if you had a general purpose media plate, a selective plate, and a differential plate, and you forget to figure out who's who, which one would tell you that it was a general purpose plate? And remember, the general purpose plate would be like for both of the organisms that grew, they would grow equally well. So make sure you understand that. Make sure you also understand what a chemostat does. It's another question that comes directly from your worksheet. So if you haven't been able to tell yet, those worksheets that we do in class for our flipped activities are very, very, very important. So make sure that you are going over those in your test preparation. So remember that chemostat is that continuous culture system, and it gets rid of the dead cells and it puts new fresh things, fresh food and resources into the environment that understand how that works by getting rid of the bad, old, icky stuff and putting fresh stuff in there to prolong the stationary phase. Also, make sure that you understand, and there's definitely a question on this, I know for sure, about all these different trophes here. So the autotroph, heterotroph, the chemoheterotroph, the phototroph. Make sure you know who does what, so you know those definitions really well. Also know those different ways that organisms can tolerate oxygen. So we talked about having that enzyme superoxide dismutase, that's a way to tolerate oxygen. We also talked about having that enzyme peroxidase that would break down hydrogen peroxide that's made as a byproduct after superoxide dismutase has been used. So peroxidase will break down hydrogen peroxide. We talked about catalase, that is also another enzyme that will break down hydrogen peroxide. So make sure you know all three, superoxide dismutase, peroxidase, and catalase, that those are all ways that organisms deal with oxygen in its toxic form. Make sure you understand those thioglycolate tubes and how the oxygen concentration decreases as you go to the bottom. So obligate anaerobes would be at the bottom, obligate aerobes would be at the top, and then kind of growing at different places in between, you have those facultative anaerobes or those aerotolerants. But big things to remember: at the top, most oxygen, obligate aerobes like that, obligate anaerobes like the bottom, less oxygen. Facultative anaerobes, they'll have more growth at the top, but you'll see them growing throughout the tube because they can deal with oxygen if it's present, and they actually make more energy from oxygen if it's present than they do if it's not there. If it's not there, it's like, okay, cool, I can survive as well. So that's why they're called facultative anaerobes. Make sure you know the difference between an obligate anaerobe, a facultative anaerobe, and a obligate anaerobe. I hope I said that right. So obligate aerobe, obligate anaerobe, and facultative anaerobe. Obligate aerobe has to have oxygen. A facultative anaerobe means it can deal with it if it's there. If it's not there, then it can deal with that too. It actually prefers oxygen, makes more energy that way. And then an obligate anaerobe, oxygen is absolutely toxic for it. Those three enzymes we talked about, superoxide dismutase, peroxidase, and catalase, are not available in those bacteria, so as a result, they don't tolerate oxygen very well. Understand those different philes that we talk about. So you've got like mesophiles and hyperthermophiles, and I feel like there should be a question on here somewhere. And if there isn't one on this, we also have, still sharing my desktop with you all. Let's do this. On Blackboard, we have in our exam review folder, This isn't your class, but still have some clicker questions that are there and more questions. So there's several questions. So it's in there, one of them. Okay. All right, so make sure you understand all of those different thermophiles, mesophiles, hyperthermophiles, and what their temperature requirements are. Also make sure that you can do these calculations for generation time. So make sure you know those formulas. And I'm almost positive I have one of those questions here. And we do. So make sure that you can do these different types of questions here. So let's just say if how long is the generation time? So make sure you know that the log at the beginning, the log of the end of cells minus the log of the beginning of cells divided by 0.301 is how we're able to calculate the number of generations. And how we calculate generation time is time divided by the number of generations, and then we also have hours in here. So make sure you remember those formulas for that, because you will have a couple of questions, and I will allow you all to use your calculators for that question there to do those types of questions. And some of those questions may come directly from the worksheets that we did in class over Chapter 6. Also, make sure you know what those different types of media are, like McConkey's agar can be both selective and differential. Same thing with MSA or mannitol salt agar can be selective and differential. Make sure you understand the growth curves that we have. First, we have the lag phase, where we don't have a whole lot of great exponential growth happening, but the cells are repping up and preparing to grow. And then we have the log phase, where we have lots of growth that's happening, and we're climbing up. Those cell numbers are increasing dramatically. And then we have our stationary phase, and at the stationary phase, the number of cells entering the population is roughly equal to the number of cells that are leaving out of the population. And then we have the death phase, where we have the rapid decline of the cells because there's no more room, there are no more resources, and there's a lot of waste that's accumulating. So make sure if you see that graph, you're able to answer questions on the lag, log, stationary, and death phase on there. And this is a question that we talked about and you've had on your worksheets again. So I think that is pretty much a good chunk of what you have. Make sure you know what an Okazaki fragment is. Remember, those are the DNA fragments on the lagging strand of the DNA. Make sure that you know who the final electron acceptor is in aerobic cellular respiration. I believe I said that before. Make sure you can do those calculations where you calculate generation time and number of generations. And also remember this, I don't think I have it in this document, but remember 2 raised to the nth power. So let me see if I can make that a superscript. Font. Let's go super, oh, there it is, superscript. Where n is the number of generations, right? Let's not have this as superscript. All apologies, good folks. Keep in mind, like I said, it's almost 2 o'clock in the morning here. So we're in as the number of generations. So if I said that, let's just go with number 39 here. 3 cells with a generation time, okay, how many cells? So 3 cells with a generation time of 60 minutes are inoculated into a culture. How many cells are there after 5 hours? Okay, so we can figure out how many generations we have here, even though the question did not give that to us. Why? Because it takes 60 minutes to get a new generation. We let them do their thing for 5 hours, so we have 5 generations that we're working with there, right? So we're going to take 2 raised to the, and I am going to be real for real with you guys. I don't know where my calculator is on my Mac, so I'm using my iPhone. because I know where it is, but it's like so far away that it's not even worth trying to find right now. So we have 2 raised to the 5th power, and that's going to equal 32. But that can't be our final answer because we started with how many cells? 3. So 32 times 3 gives us 96. Correct answer, 96. So also make sure you remember this whole thing that we did here. So once again, if that did not make sense, please rewind this video and go back to it again. So you're going to have a couple of questions with that math. So make sure you understand it and make sure you can do it. Make sure you can answer a short answer question on explaining those steps in aerobic cellular respiration, starting with glycolysis, the Krebs cycle, and the citric acid cycle on there. Make sure that you can do that. Also, make sure you can explain Griffith's experiment, like what is transformation? Why did he use the R strain and the S strain? What was the heat-killed S strain? Why were the results unexpected? So just describe the entire experiment. And you have a couple of questions on your last Chapter 7's worksheet that went over just that talked about his experiment in great detail. And there's a really awesome video that we posted on Chapter 7 that talks about that. So I am fried right now, like literally, so that is where I'm going to end things for the day. If you have any questions, please do not hesitate to email me, and we will get you straightened out. We'll get you where you need to be. So have a great morning, noon, or night, and I will see you all very soon. Bye.